Coming to the unit 2, the first topic is about drugs and drug targets and receptors. Generally, drugs produce their therapeutic effects by producing biochemical and physical changes in the target tissues. These tissues of the host and also the organisms which invade the host. They changes due to physical and chemical properties of a drug action on the drug targets. These are namely the receptors, enzymes, carrier molecules, ion channels. To get the drug action, it is essential that surface concentration of drug that reaches the site of action, that means the bioavailability, and remains there for a sufficient duration. How much duration it is staying in the body, we have to figure it out. And the tissue is susceptible for the drug action. And also the magnitude of drug action is proportional to the concentration of drug at the site of action. It should not be too much heavy and it should not be too much low. Understanding target receptor is nothing but your macromolecule mechanism, and it is very important to understand the action and also the effect of the drug. You have to know the biochemical pathway. Receptor component of a cell or organism interacts with a drug and initiates the chain of a biochemical events, various biochemical events, and these the steps which are involved in biochemical events leading to the drugs observed effects. They have specific binding sites that are definite in size and shape. Most are present on near the membrane. Some lie in the enzymes or genes. You can see Langley and Ehrlich, they introduced the concept of receptor. The Langley from 1852 to 1895. He studied the effects of atropine against pilocarpine induced salivation in cats. And he postulated that there was a receptive substance in the nerve ending or a gland cell with which both atropine and pilocarpine are capable of forming these compounds. And Ehrlich observed that certain dyestuffs acted selectively, staining some cells more deeply or in different way from other cells, and suggested that drugs with selective actions on particular cells could be developed. You can see the four major families of receptors. One is ligand gated ion channels. We take example nicotinic ion channel, and GPCRs, nothing but G protein coupled receptors. We take alpha adrenoreceptor and enzyme linked receptors. For example, insulin receptors, nuclear receptors. For example, glucocorticoid receptors. All these receptors, these are the main important four receptor families, and receptors that do not fall in these four receptor families are specific ion pumps. You might have studied in the cell biology class that these ion pumps, sodium, potassium and ATPase specific enzyme pumps. Example, the 5-phosphodiesterase pumps, and structural proteins such as Golgi into tubulin and cytosolic proteins, cyclospore into immunophilins. These are the examples. And there are four main types of receptors. And the one is the first type. First type is type 1 ligand ion gated channels. These type 1 ligand gated ion channels couple directly to membrane ion channels. Whenever the agonistic molecule binds to it, it opens the channel and causes depolarization, or we can call it hyperpolarization. This will cause the change in cytosolic ionic composition depending on the ion that flows through. And the channels control the fastest synaptic events. The synapse you can see in nervous system. It controls the fastest synaptic events in nervous system. And excitatory neurotransmitters such as ACH hormone, glutamate, it causes an increase in sodium and potassium pumps and sodium potassium permeability that results in a net inward current and depolarize the cell that could generate an action potential, nerve potential. Examples, nicotinic receptor, GABA receptor, glycine inhibitor receptor, 5-HT receptors. Signal molecule, whenever the signal molecules comes and bind to a receptor, specific receptor at a specific site, that means active site, it leads to a conformational change and it opens the channel and allows the ions to flow through. There will be the electrical potential generates between outside membrane and inside membrane. It flows into the cell and the change in the ion concentration, this triggers the cellular response. So out of the four, the second most important receptor is type 2 G protein coupled receptors. And these are also called as 7-alpha helic membrane spanning around the hydrophobic amino acid segments, and it runs to 3 extracellular and 3 intracellular groups. Binding of these mediator molecules induces a change in the conformation and unable to interact with the G protein that lies at the inner leaf of the plasma lemma. The G protein consists of 3 subunits, alpha, beta and gamma. In the inactive state, GDP is bound to alpha subunit and leads to activation. Further, it leads to displacement of GDP by GTP. Activated G alpha GTP dissociates from beta and gamma subunits, then associates with an effector protein and it can alter its functional state, and also it could control all the cellular events and could control the functions. If you see, the functions of certain receptors are ligand binding, message propagation through signaling, and functional domains within the receptor. Ligand binding domains spatial and energetically suitable for the binding of the specific ligand, and effector domain. This effector domain which undergoes a functional conformational change. A receptor effectors, a receptor may be exerted directly on its cellular target, or effector proteins are conveyed by intermediate cellular signaling. These are called transducers. In conclusion, receptors largely determine the quantitative relationship between concentration of the drug and pharmacologic effect, and it is responsible for the selectivity of drug action. It mediates the action of agonist and antagonist.